Assalamualaikum and have a good day. Our group will dive into the fascinating finding from the article Introbacterial Growth Promotion by Intestinal Acylcarnitine, a biomarker of dysbiosis in inflammatory bowel disease. Will uncover how intestinal acylcarnitine promote the growth of enterobacteriae, serving as a biomarker of dysbiosis in inflammatory bowel disease.
This study reveals that Enterobacteriaceae like E. coli consume carnitine and acylcarnitine, increasing their numbers and causing gut dysbiosis. Different bacteria have distinct consumption patterns. For example, E. coli consume carnitine while P. Pseudomonas aeruginosa consume acetylcarnitine. Also, the correlation between IBD and carnitine and acylcarnitine level is that higher fecal carnitine levels influenced by a Western diet rich in red meat are associated with increased E. coli levels, positively correlated with pediatric IBD. The hypothesis driving this research is that by understanding the interplay between gut microbiota and its metabolic products, such as carnitine and acylcarnitine, we can clarify the mechanisms causing IBD. The gut microbiota plays a critical role in the pathophysiology of IBD. The objective of this research are to investigate the consumption of carnitine and acylcarnitine by gut bacteria, to assess the impact of carnitine and acylcarnitine on gut microbiota, and to investigate the relationship between carnitine and acylcarnitine to IBD. The major method used to complete this study is firstly, fecal metabolomic analysis, which identify and quantify the levels of carnitine and acylcarnitine in fecal samples from pediatric IBD patients and healthy controls. Secondly, Citrobacter rodentium colitis model to induce colitis in mice and study the effects of acylcarnitine on the gut microbiota. And lastly, growth curve and anaerobic culture, which determine the growth of various bacterial strains in different conditions, including the presence of carnitine and acylcarnitine.
So we are continuing in the result part. The result obtained, the carnitine and acylcarnitine are correlated with dysbiosis in pediatric intestinal bowel disease, which have negative correlate with the most bacteria but positively correlate with the Enterobacteriaceae like E. coli. Next, microbiome depletion associated with higher luminal carnitine and acylcarnitine, which indicate that microbes decrease and the levels carnitine and acylcarnitine increase in the intestinal domains. Next, in a mouse model of the colitis that having an inflammatory condition of the colon, the levels of the carnitine and acylcarnitine will be increased, and the increase not limit to the intestinal tracts, but also found in the bile and plasma of the mice. Finally, dietary supplementation with acylcarnitine promote the growth of pathogens like Citrobacter rodentium in mice.
Host interactions with gut microbiota significantly change during intestinal inflammation through alteration in the metabolome. Dysbiosis in IBD leads to reduced level of secondary bile acid and short-chain fatty acid due to the loss of Clostridium species. Interestingly, IBD patients show increased fecal level of carnitine and acylcarnitine, which are primarily delivered from the host rather than produced by bacteria. Carnitine, a quaternary amine, is utilized by bacteria as a carbon and nitrogen source, osmoprotectant, or electron acceptor. Elevated level of carnitine and acylcarnitine in fecal sample from IBD patients have been linked to a higher abundance of Enterobacteriaceae, particularly E. coli. E. coli utilize carnitine and acylcarnitine as energy source and result in mitochondrial dysfunction in the gut epithelium, promoting their growth. This process exacerbates dysbiosis, shifting the balance away from beneficial bacteria like Firmicutes and Bacteroides. The consumption of red meat, a rich source of carnitine, is associated with higher fecal carnitine level and a greater prevalence of IBD, suggesting dietary influences on disease progression. In contrast, plant-based diets are linked to a reduced risk of IBD because plants have level of acylcarnitine 2 to 3 lots lower than animal products.
In conclusion, the study revealed that high gut carnitine and acylcarnitine level are linked to dysbiosis and enterobacterial proliferation, particularly in individuals with IBD. Metabolite will serve as biomarker for early diagnosis and disease monitoring, and as therapeutic target for managing IBD symptoms by modulating this metabolite or targeting specific metabolite pathway. Next, we can analyze and agree with the author's conclusion. The study revealed a positive correlation between fecal carnitine and acylcarnitine level and enterobacteria abundance in IBD, supported by robust methodologies like mass spectrometry and microbiome analysis. Colitis and intestinal damage increase luminal carnitine and acylcarnitine in the gut lumen, with enterobacteria consuming this metabolite in a taxon-specific pattern supported by in vitro and in vivo experiment. The article explored carnitine metabolism's impact on the gut microbiome, comparing findings with existing literature, highlighting its role in microbial growth and its implication for IBD. The author acknowledged and incorporated related studies finding for a comprehensive view, promoting collaboration and data sharing with other research programs to integrate diverse insights.
This about the research limitation. The study was specific in investigate individual with pediatric intestinal bowel disease. This limit the result applied to the adult. Next, the use of the in vitro and animal models in the experiment may not show the accurate result. Why? Because humans have the complicated gut environment. Therefore, it is essential to verify the finding through the human clinical trials. That's all from us. Thank you for your attention.